Okay, so Wednesday we looked at the structure of DNA, and some of you extracted DNA on Wednesday, some of you will extract DNA on Friday. We're going to kind of go through, I don't really care that you can read through the objectives if you want to. We are going to look at how we use this in forensic science. So again, a review, DNA is a double helix, two-coiled DNA strands. It is composed of nucleotides, which contain a sugar molecule, deoxyribose, a phosphate group, and a nitrogen-containing base. In humans, the order of the bases is 99% the same. So we have 4 basic bases: adenine, cytosine, guanine, and thymine. We talked about before, adenine always bonds with thymine, cytosine always bonds with guanine. Genes are the portion of the DNA that codes for proteins. It's found in all of your nucleated body cells, so white blood cells, semen, saliva, urine, hair roots, teeth, bone, and tissue. It is not found in red blood cells because red blood cells do not have a nucleus. They are most abundant in your buccal or your cheek cells, and red blood cells have no nuclei, therefore they cannot use those for testing. So when they do use DNA testing from blood, they're testing the white blood cells and not the red blood cells. So when you're talking about DNA typing, this is a method in which DNA is converted into a series of bands that will distinguish one individual from another. We also call this DNA fingerprinting. Basically what they're doing is they are cutting pieces of DNA with enzymes to cut it in certain points on the DNA molecule, and each individual will have different size molecules after it's cut by the enzyme. So only 1/10 of a single percent of DNA, about 3 million bases, differs from one person to the next. So scientists use these regions to generate a DNA profile for an individual. So they target the areas, the 3 million bases that they know are different between one another. So you have coding and non-coding regions. So 3% of human DNA codes for protein. The rest of it is non-coding. It's repetitive. It has the same repeating sequences over and over again. Difference is we differ in the number of times it actually repeats. So 50% of your human genome or DNA has these repetitive sequences. So when we are using DNA profiling, we are using this to identify potential suspects. We're also using this to exonerate or free individuals, and we saw that with the Innocence Project, that a lot of individuals who were put in prison on hair or fiber alone and not really having the technology at the time, a lot of them are using DNA evidence now to go back and exonerate them, identify crime and casualty victims. So if they are not physically able to be identified, you can use their DNA. It's also used in paternity testing to see who is the dad. We use it now to match organ donors. So the more, besides just blood type in common, the more genes you have in common on the organs that you are donating, the less likely it is that you will reject the organ. So there are some things that we do in the process of DNA fingerprinting. There is what's first called restriction fragment length polymorphism. These are all big words. Polymerase chain reaction, short tandem repeats. Whoops. So basically, first step we do is we use restriction enzymes to cut DNA into smaller fragments. Those can then be separated and characterized for identification. So if we cut DNA with certain enzymes, we will get different patterns for different individuals. Then they run them on what we call a gel, and that gel separates the DNA by size. So you end up with a fingerprint or pattern for each individual depending on the size of their fragments. The shorter fragments go faster, and this gel is sitting in a liquid and has electricity running across it with a battery. So those that are the shortest segments go the fastest, those are the longest segments go slow. So in order to do this, number one, we have to separate DNA from the cell. which we did that in class this week. Second thing you do is you cut it with restriction enzymes. So you are cutting it at known base sequences to get shorter strands of DNA. Third thing you do is you sort them by size using electrophoresis. And then the fourth thing you do is you analyze the specific alleles for identification. So you're looking at the patterns to see if the patterns actually match or not. Every individual has their own pattern for DNA except for identical twins. Identical twins are the only ones that cannot be separated or individualized using DNA analysis. So to give you an idea, we have sets of enzymes that we know where they cut on the DNA. So if I use BAMH1, it cuts on this sequence between the G and the G. HEY3 cuts between the C and the G. PST1 cuts between the A and the G. And BGL2 cuts between the C and the T. I do not expect you to know that. We're going to go through several examples of these and show you how to cut the DNA fragments, or how the enzymes cut the DNA fragments in order to determine patterns or these fingerprints. So right now we are not going to go into polymerase, because polymerase really, chain reaction, is a way of amplifying it after you cut it. So we cut the DNA, we get the fragments, and so what we do is we go through a process that multiplies the amount of DNA that you have, so you have a large enough amount to run on a gel. So we're going to stop here because I want you to understand how the cutting of the DNA works. Once we do a couple of exercises on this, then we will go into a little bit more about PCR and what the tandem repeats are.